Recombination. Recombination is a process by which pieces of DNA are broken and recombined to produce new combinations of alleles. This recombination process creates genetic diversity at the level of genes that reflects differences in the DNA sequence of different organisms. In eukaryotic cells, which are cells with a nucleus and organelles, recombination typically occurs during meiosis. Meiosis is a form of cell division that produces gametes or egg or sperm cells during the first phase of meiosis. The homologous pairs of maternal and paternal chromosomes align. During alignment, the arms of the chromosomes can overlap and temporarily fuse, causing a crossover. Crossovers result in recombination and the exchange of genetic material between the maternal and paternal chromosomes. As a result, offspring can have different combinations of genes than their parents. Genes that are located further away from the same chromosome have a greater likelihood of undergoing recombination, which means they have a greater recombination frequency. Then site-specific recombination. Site-specific recombination, also known as conservative site-specific recombination, is a type of genetic recombination in which DNA strand exchange take place between segments possessing at least a certain degree of sequence homology. Enzymes known as site-specific recombinase (SSRs) perform rearrangements of DNA segments by recognizing and binding short specific DNA sequences, sites at which they cleave the DNA backbone, exchange the two DNA helices involved, and rejoin the DNA strands. In some cases, the presence of recombinase enzyme and the recombination sites is sufficient for the reaction to proceed. In other systems, a number of accessory proteins and or accessory sites are required. Many different genome modification strategies, among this recombinase mediated cassette exchange (RMCE) and advanced approach for the targeted introduction of transcription units into predetermined genomic loci rely on SSRs. Then site-specific recombination systems are highly specific, fast and efficient even when faced with complex eukaryotic genomes. They are employed naturally in a variety of cellular processes, including bacterial genome replication, differentiation, and pathogenesis. And movement of mobile genetic elements. Site-specific recombinase, or SSRs, are enzymes that bind to the shorter DNA sequences, cleave DNA backbones, and aid in the exchange of DNA helicases. Finally, it rejoins the strands. In some cases, accessory proteins and accessory sites are also required. Site-specific recombination is a fast, specific, and efficient method. It is seen in bacterial genome replication, pathogenesis, and differentiation, and movement of mobile genetic elements. Then non-homologous recombination, enjoining, or non-homologous recombination. Non-homologous enjoining is a pathway that repairs double-stranded breaks in DNA. NHEJ is referred to as non-homologous because the break ends are directly irrigated without the need for a homologous template, in contrast to homology-directed repair, which require a homologous sequence to guide repair. So NHEJ is active in both non-dividing and proliferating cells, while HDR is not readily accessible in non-dividing cells. The term non-homologous enjoining was coined in 1996 by Harber. And NHEJ is typically guided by short homologous DNA sequences called microhomologies. This microhomologies are often present in single-stranded overhangs on the ends of double-stranded breaks. When the overhangs are perfectly compatible, NHEJ usually repairs the break accurately. An impious repair leading to loss of nucleotides can also occur, but is much more common when the exchanges are not compatible. Inappropriate NHEJ. So, NHEJ usually repairs a break accurately. Imprecise repair leading to loss of nucleotides can also occur, but is much more common and the overhangs are not compatible. Inappropriate NHEJ can lead to translocations and telomere fusion, hallmarks of tumor cells. NHEJ implementations are understood to have been existent throughout nearly all biological systems, and it is the predominant double-strand break repair pathway in mammalian cells. In budding yeast, Saccharomyces cerevisiae. However, homologous recombination dominates when the organism is grown under common laboratory conditions. And homologous recombination is a type of genetic recombination in which nucleotide sequences are exchanged between two similar or identical molecules of DNA. During the formation of egg and sperm cells or meiosis, paired chromosomes from male and female parents align so that similar DNA sequences can cross over or be exchanged from one chromosome to the other. This exchanging of DNA is an important source of genomic variation seen among offspring. This is a type of genetic recombination that occurs during the formation of egg and sperm cells. During this process, meiosis, what happens is that paired chromosomes from the male and female parent align so that similar DNA sequences from the paired chromosomes have an opportunity to flip flop or cross over one another. This crossing results in the shuffling of genetic material, just like some gentle shuffling of a deck of cards. And it's important because it's one of the sources of genetic variation that we see among offsprings of a set of parents. This offspring receives bits of DNA from each of their parents, but also from their grandparents and ancestors. In bacteria, homologous recombination is a major mechanism of DNA repair and facilitates the incorporation into DNA of genetic material received via horizontal gene transfer and transformation. In viruses, homologous recombination helps shape viral evolution. Here you could see site of crossover, homologous chromosomes, then non-sister chromatids recombined genetic information. Next is about homologous recombination, site-specific recombination. Definition: homologous recombination is a mechanism of exchange of genetic material between two identical DNA or RNA strands. Site-specific recombination is the exchange of genetic material between DNA strands that possess a certain level of sequence homology. Length of the strand: the exchange occurs between long DNA strands. The exchange occurs between short DNA strands between 30 to 200 nucleotides in length also. Location: it occurs anywhere in the homology. It occurs only at specific sites. This is a comparison actually. Site-specific recombination is represented within this blue ink colored. So then homologous recombination is the mechanism of exchange of genetic material between two identical DNA or RNA strands. The exchange occurs between long DNA strands. It occurs anywhere in the homology. The enzyme machinery is relatively simple and small for homologous recombination. Recombination of gametes during meiosis, horizontal gene transfer in bacteria and virus, bacterial genome replication, differentiation, pathogenesis. Then homologous recombinations. And site-specific recombination is the exchange of genetic material between DNA strands that possess a certain level of sequence homology. Then the exchange occurs between short DNA strands between 30 to 200 nucleotides in length. It occurs only at specific sites. It occurs specific enzyme and enzyme machinery for recombination. Then movement of mobile genetic elements.